As students in the Science in the Bay PBL class, we work in groups to solve real-life situations and get first-person experience. Recently, we were presented with this project called Panic at the Cisco. It was structured around the fact that we had to learn about genetics and natural selection. We were introduced to what was referred to as the creatinine problem.
Fisheries biologists have noticed that the morphology, the trait variation has increased in the Ciscos of the Great Lakes over time, making taxonomy very difficult.
Researchers began to struggle to set the different fish apart. So we researched and looked into 3 of the 5 variations of the Cisco species, and each group focused on a single feature in fish. From there, we gathered information and formed claims as to why these changes occurred.
So the way we did this project was pretty unique due to remote learning, and it was definitely challenging because we couldn't be in class and we had to do it on our own time. So the way we did it is we downloaded an app called Pixel Zoomer on the Chromebook, which this is where we could take a screenshot of the fish and measure from one end to the next, which then we could plug into a spreadsheet. And our goal was trying to help the scientists what caused variation, like human impact, climate change, pollution, anything.
We were remote for the entirety of this project, so students would come to class and they would break out and do some measuring and then take a look at some of the ratios and come up with problems or maybe reasons why these characteristics or these ratios would be different.
As individuals, we were assigned a Cisco trait and constructed a CER as an end result of our project. We used our knowledge to make a claim, supported it with our data from our spreadsheets for evidence, and elaborated with reasoning. We learned how to take our knowledge and apply it in a professional CER format. From there, we also set up future years of our class with our data and our CERs.
Throughout our Panic at the Cisco project, we've had a lot of help from our community partners. This project would not have been possible without them. Fishery biologists Chris Olds and Brandon Schroeder have aided us throughout the whole process. They helped us analyze the data from the fish and understand what the data we gathered meant. They also answered any questions we had about anything to do with the project and cleared up many misconceptions. They were a big part of the project and were a lot of help throughout the whole process.
I have had the opportunity to work with the Alpena High School Science in the Bay initiative to provide some Alpena High School genetics classes with a science project that's hands-on and a real-world problem that we're facing right here on the Great Lakes right now. We've been collecting these fish for about a decade now and taking digital images and measuring their structures. So things like their dorsal height, their body depth, their head length, and coming up with ratios that we can compare each species across the Great Lakes and then also within a species of the same form. And what's really been exciting is to be able to take that data and those images and share them with the students and let them do the exact same thing. Take these images, take measurements, and then compare them across the species and look for differences and ask, using the scientific method, ask questions about why they're different and how this has been occurring. And so to see their wheels turn and think critically about these questions that we're facing every day as scientists has been just fantastic.
What was really fun for us was some of our students thought outside the box and asked questions that even our scientists didn't quite know the answer to, which made them think maybe they need to look at some of those factors and why the characteristics of the fish are different.
Although we had to work on a majority of it from home, with Google Meets and mentor guidance, we were able to learn and participate in this amazing experience and be part of real-life experiments that will hopefully continue to expand in future classes.